We see in a number of studies that below ground inputs to soils are much more likely to be retained. So soil carbon is initially mostly root carbon, not everything that we harvest and see above ground. And that's a little bit of a frame shift from maybe what I was taught when I was in graduate school, that, you know, it's above ground litter and, you know, litter bag studies for decades. Really, most of that carbon is respired back to the atmosphere. So it's the carbon that's literally inserted into the ground in roots that has a greater chance of staying and sticking. What causes it to persist? If you're not part of the soil sphere, then you may not know that about 10, 12 years ago, that we just had a complete kind of revolution in our thinking about what makes organic carbon stay in the ground. And it's really not anything to do with its recalcitrance. So nothing to do with how aromatic it is, or if it's hydrocarbon, or if they're lipids. It's not about the chemical weight. It has a lot more to do with the place and space and time and other actors that are coincident with that carbon. So that's a frame shift. It's not just old carbon is not necessarily just the kind of gnarly stuff that nobody wanted to eat. A lot of the carbon that we do see persisting in soil, and when we talk about persisting, I mean that it has a longevity that is decades to millennia, and we see that in our radiocarbon analysis. That organic matter can often look like polysaccharides, and we presume that comes from EPS that either roots or microbes have produced. We also have good evidence that all of those kind of sticky goo compounds in EPS are very good at aggregating soils and making little packets and literally physically protect organic matter. So I just wanted to end with saying that, you know, we have some evidence that that's one way that carbon becomes stabilized. There are still other mechanisms that we're, to be blunt, not good at explaining. That said, we have sort of global evidence to suggest that deep root carbon is persisting. If you look at databases where we've looked at the age of carbon throughout the globe, the places where we tend to have the deep carbon, maybe on the old prairie mollisols in the US and in Russia, those are sites where we tend to have higher, not only stocks, but carbon that is sticking around for a long time. And interestingly enough, we tend to see that more often in grasslands than we do in forests. And in an analysis a postdoc of mine, Cesar Terrer, did a couple of years ago, he looked across a number of different elevated CO2 studies where essentially plants are being fertilized in controlled environments, either in the lab or out in the field, and showed that when you give plants the ability to fix more CO2 and create more photosynthate, we actually tend to see carbon being stored below ground in grasslands. Whereas in forests, it's all in the bowl of the trees and mostly above ground. That's fine, that's a great place to store carbon, unless you live in the West and we have, you know, serious concerns about fire. So I have primarily studied grasslands, not necessarily because of this, but that it's kind of reinforcing of a lot of our efforts to think about carbon below ground as a sort of safe place in grassland systems. There's also a number of studies, this is some data from a European study that looked at all the different factors that were likely to increase soil stocks, and really the top factor was maximizing root biomass. So in my group, the way that we study this, at least very kind of mechanistically, is through something called isotope tracing or stable isotope probing. Those are kind of related techniques. We do this both in the lab and in the field. What you're looking at in the photo on the right-hand side is our greenhouse at UC Berkeley, where we were growing switchgrass in native marginal soils from southern Oklahoma. We had recreated a meter-deep soil profile and grew these plants that we had sequenced and had a nice clonal singular genotype in an atmosphere that was filled with 13 CO2. So it's the expensive kind of CO2, right? It's the CO2 that allows us to trace that unique heavy isotope as it's not only being fixed by the plant, but exuded into the rhizosphere, taken up by microbes, sorbed onto minerals, that whole kind of pathway. So that's something that we do in almost all our experiments. But I'm going to start actually here in the field. This is our one of our sites in southern Oklahoma, where we've been looking at a series of switchgrass plots. Some of these are up to 10 years old. So on the right-hand side, that's a 10-year-old alum field, and on the left is a field that's been in shallow-rooted annuals, soy and wheat and some corn, and in a sort of standard rotation. And we compared the soil stocks between the sites where we have these deep-rooted perennial grasses, switchgrass, with the annuals, thinking maybe if switchgrass is truly pumping more carbon in the ground, we'll see a difference. And here's some of the data. So in 10 years, at this particular site, this is in Stillwater, which is actually the middle of the state, we actually saw almost a doubling of soil carbon. So the switchgrass soil data is in blue, annuals is in red. And you can see we went down to 3 meters. The majority of the shift is actually kind of above 2 meters, but it's statistically significant all the way down. So we see that in the total carbon. And then when we looked at the age of that carbon, so we used our ability at Livermore, we have a Center for Accelerator Mass Spectrometry, the ability to measure radiocarbon as a way of dating and looking at turnover time. You can see that the blue data is shifted towards 0. That means it's more modern. And that makes sense in that you've got plants that are fixing modern CO2 and stuffing it in the ground. You've got still a lot of background of older carbon, but you can see that they're moving it towards the modern numbers simply because of the new photosynthetic that's being put below ground. So that's exciting. That's what we would have expected. And those are 2 kind of orthogonal pieces of data that suggest, yes, these deep-rooted grasses are putting carbon in the ground. In addition, in both that greenhouse experiment that I showed you a second ago with the columns of soil and in the field, we measured extracellular polysaccharides, EPS, and did our best to distinguish between plant and microbial EPS. That's a little easier said than done, but regardless, we're able to look at least at the total amount of EPS, and you can see in the blue data, there tends to be more all the way down the profile. And this is not only just around the root, this is kind of throughout all of the soil. I can't make a direct connection that EPS is why we're seeing more carbon, but it certainly makes sense to me. We've got more of this sort of sticky goo being exuded from roots, and we see more aggregation. I don't have that data up here, but we see more water-soluble aggregates, little particles of soil holding together. And then we're seeing more of EPS. So we think that those things are likely connected. We're kind of packing things together.
I wanted to ask about the sort of deep-rooted perennials in particular. So my agronomist friends always remind me that like the plants don't just grow their roots like really deep for fun, that like they go in search of water.
Yes, yes.
So it's just very easy for me to forget that like they're like, look, what's happening with water? Like what is there is there a water table that they're sort of reaching for or something that can impact how deep the root systems are?
Yeah.
I mean, did you see differences here? Like, has that been a problem in some of these field experiments?
So that was not a planted question, and I sometimes I thank you for asking. I sometimes show this figure with actually one additional data slide. I just didn't want to kind of inundate you with too much, but if you can imagine one more slide here that actually shows the 18O isotopes of water with depth that we looked at in the soil, but also in roots. What that data shows is the deep roots are moving a bolus of soil water that's really quite deep up into the surface. They are doing their job. And yes, so in this system particularly, those roots are looking, the deepest ones in particular, are looking for water, and they are shifting it from, not an aquifer level, but deep in the soil horizon up to the surface. And we don't see evidence of that in the annuals because the roots don't go that deep. That said, I'm going to give you a big but I'll start with it right now. This is one site. These happen to be soils that they are mollisols, but just barely. The carbon is really on the edge of what would typically be called a mollisol, but they are very deeply weathered. There are many, many other sites that we've been to since where there are hard pans, there are rocks, there are all sorts of other geomorphological reasons why the roots can't get down into accessing the deep water. And there's places like Illinois or you don't need to. It's, the water is kind of coming from the sky and you don't need to be reaching down into those deeper regions. So in some cases, that could be why we're not seeing carbon being deposited. That is simply those roots don't need to go looking that deep. Yes, so great question. So speaking of which, we did replicate this study across a whole bunch of different sites across the US. So we literally got in a van, had a trailer with a geoprobe, and drove all over the eastern Midwest. And I don't know if you could see the little blue stars there, but we went to Texas, Oklahoma, Mississippi, Alabama, North Carolina, New York, Illinois, South Dakota,
All the hotspots.
all the hotspots, Wisconsin, we went to, and Michigan, ABS. And we were able to make contact with a number of different landowners who had similar situations. They had planted switchgrass back in the aughts when, it's going to save the world, we're all going to plant switchgrass. And they still had those fields. In many cases, nothing had been done other than some, harvesting of above ground biomass. And we compared it to, as best we could, the next door field that had been in some sort of annual rotation. And here's the data. It's, I'll just say from the outside, this is a little bit of a complicated plot, but here I'm conflating the total carbon data and the 14C based data. Again, those being two kind of orthogonal ways that we can understand whether carbon is accruing. If it's getting younger, and if we actually have more of it, and we have a sense that, these roots are putting more carbon in the ground. So if you're in the upper right SOC gain gray part of the plot, the soil is ostensibly gaining organic matter over the 10 years or give or take, that these plants have been in place. What I think you can first see here is not all the data points are in that area. There were lots of places where we actually did not see an increase in carbon statistically, and in some cases actually had seen a decrease. There also tended to be an association of the places where we did see carbon increasing, that the soils had started off with less carbon to begin with. They tend to be the southern sites. If you can see the smaller circles, those are in the southern US. They tend to be on the more poor, what we sometimes call marginal or highly weathered soils that sometimes are more water limited as well. So is there a relationship there between the soils that start off with less carbon and their ability to gain carbon? So that steps into a bit of a debate that's been going on for a couple of decades about so-called carbon saturation. Is soil essentially a sponge that can keep holding and holding and holding more and more carbon until it hits kind of a threshold, and then it literally starts dripping out the bottom? I don't know if that analogy really works for you, but I've always thought it's kind of a fun idea that they literally just absorbing and absorbing. And there is evidence to suggest that there is an asymptote that soils can hit, and after which they really can't hold more unless you're in, a frozen tundra and you're a histasol. That's a kind of different scenario. There is also data, this is another post-hoc study with Kat Georgiou in my group, who show that soils that are starting at only 10% saturation, so the sponge is pretty dry to begin with, is able to accrue 3 times faster than a soil that started with more carbon. So cool, that suggests we should go plant switchgrass in the southern US, done. However, right, I like to tell stories this way, if you don't mind. We've actually just done a really deep dive on some of the statistics in these data sets, because it turns out that when you're looking at the change in carbon across a site, whether it's a time zero versus 10 years later, or like we did, a space for time substitution, where we have an annual and a perennial plot that are right next door. And you're always referencing that back to whatever the initial carbon was, which means you've got, essentially think of it this way, correlation between your axes. And you have a problem that's called reduction to the mean when you plot the data. The initial data and the sites where you have low carbon have a high likelihood of having a lot of spread in the data, a lot of variance in the data. And you, through random error, can get an association that looks like you're increasing carbon, when the reality is it's a statistical effect. So we just published that paper. Now we can go back to the actual data and we can do a correction And essentially, that paper is not out yet, but the short end of the story is, be careful when you see people claiming that they're seeing a soil carbon increase. There can be reasons that have to do with the presentation and the way that it was analyzed. The other thing that limits the amount of carbon that we can pull below ground is simply how much photosynthesis there is, right? And there's been a wonderful analysis of this by Henry Jansen that came out this past year that suggests, some of the numbers that people are floating around, gigatons of carbon stored in the US by 2050 are just not possible given the constraints on how much photosynthesis and how much land we have. So this is kind of meant to be a little bit of a cautionary tale of, yes, we can put carbon in the ground, but there are some limits on how fast that can happen and how much we really can expect. I'll end that part of the talk by just saying we're trying to go around those limits in a new project that I'm part of at the Innovative Genomics Institute, which is associated with UC Berkeley. This is funded by the Chan Zuckerberg Initiative. We're actually trying to combine some CRISPR editing of the photosynthetic capacity of plants to essentially make them more efficient with some editing, which is creating cultivars that have more deep roots and more production of that sticky goo, the EPS, and with myself doing soil carbon measurements. So we're trying to actually think about creating plants that are more designed for putting carbon in the ground. So stay tuned. It'll be probably another 5 years before we get any new data on this, but that's in progress. Okay, so that was all kind of about roots. I want to talk a little bit about microbiomes because roots don't just shunt their carbon directly onto mineral surfaces, that there's an intermediate there. Yeah.
And maybe you're going to cover it, but the point of getting more EPS is just to increase the amount of necromass, because in theory, like necromass is that carbon hangs around for longer.
The point of the EPS is a little unclear. Mechanistically, my simple brain thinks sticky holds aggregates together, they literally clump more. We have lots and lots of data that shows when we have better clumped soil, better aggregated soil, we tend to hold more carbon. So is it really about the physical packing together of a donut that holds carbon in the middle and that's the jelly, or is it about some other more complicated mechanism that I don't know yet? But so I think I mentioned the word necromass. I don't know if that's in your common parlance. I just love it. I love giving this, talk on Friday the 13th, which I've done many times, and I kind of give it a whole spin about death in the microbiome. A lot of soil organic matter is very likely made of the bodies and body parts of microorganisms. Something like only 5% of microbial biomass in soil is actually alive. Most of it's kaput, and that's really good. And we want more of that turnover because each time that wheel spins, we think some of the carbon being caught and trapped in monolith pools. Here you're just looking at what cells look like on the surface, whether they're bacteria or some fungal hyphae. And every time I get on the SEM and take these kind of images, I'm just seeing all kinds of detritus. And sometimes you can tell what it was to begin with, that it was part of a cell or a diatom or protists even. And sometimes it's just guts and you don't know. But we have made a long list of different traits, both the biochemistry, the lifestyle, the stress tolerance, all sorts of things that could be traits that are important for predicting soil carbon. We are not trying to measure every single one of these traits. I think that would be a life's work, but we're really trying to figure out, are there maybe a top 3 or 5 amongst this list of traits, which is really good at predicting soil carbon? And could we maybe then, in some magical world, enrich for those kinds of organisms or those kinds of traits? And again, a lot of the way that we're doing that work, this is for the SFA project, is through stabilized tracing. I mentioned earlier, I showed you those deep switchgrass pots. We also do it in some more annual grasses and then even out in the field. So these are all different ways where we can essentially expose a microbial community to an isotope label. And that could be 13C by a plant pumping in the ground. It also is sometimes 18 in water, which just tells us, are you awake? Are you active? And I'll just tell you, all these studies are telling me that it's much less than 100%. Sometimes only 8% of the total microbiome is actually active. And that, when you're putting together a genome assembled metagenome, is really important to know. I don't need to make mags of everybody. I just want the ones that are active because I care about what their biochemical function is. So that is why I am just a huge proponent of using isotope tracing to kind of narrow in on the organisms who, from my perspective, who matter. So I'll just say that there are a number of traits that we've pointed out as being important to forming carbon that seems to be mineral associated or is likely to persist. So on the y-axis here, we have 13C mineral associated soil organic carbon, MASC. It's kind of a terrible acronym, but and then on the x-axis, there's either growth rate or EPS or carbon use efficiency. And what we see, without even getting into the weeds here, is that it really kind of depends where you are. It's not the same traits all of the time that are predicting whether soil carbon is going to accrue. So in a rhizosphere where things are growing quickly and organisms are very dynamic and a lot of turnover, growth rate seems to be really important. But carbon use efficiency of an individual organism, not so much. And that actually kind of goes against what the literature has been telling us for quite some time. The detritosphere, where I think of organisms as having a little bit more time, things are more moving at a slightly more sedate rate, you've got extracellular enzymes being produced. It is both the biomass carbon, the EPS, and the carbon use efficiency that seems to predict whether carbon is becoming mineral associated. So long story short, it's not like we're going to get just 3 traits and that's going to predict carbon. It's going to really depend on where you are and what the context is. So as we try to dive deeper into understanding the kind of metabolic and genomic traits of these organisms, we're using metagenomics, again, informed by the stable isotopes. And this is just some data that we generated some years ago, actually, from one of our first studies where we looked at partial genome bins, see a huge amount of diversity, not just in bacteria, but also eukaryotes and phage. So we got really excited about phage when we started realizing that we could actually see phage in our metagenomes. This was probably 5-6 years ago now. Turns out many of the active bacteria are actually hosts for phage. And this is when we were mostly doing our mapping using CRISPR. We now have lots of ways to find phage host maps. A Burkholderia that is our most abundant organism, our fastest grower, just the winner in a rhizosphere, has got, I think, 5 different phage that want to go after it. And this is, obviously just one time point. When we went and looked at that particular phage genome, we were able to actually make a circular genome and really characterize a lot about its capabilities. We even created a proteome, and there was a lot of variance-related proteins involved in this particular organism. So, long story short, we have good evidence to suggest there is a lot of kind of predator-prey, lions and gazelles kinds of activity going on in the rhizosphere, and that fast-growing organisms are just as quickly being targeted by phage and very likely laced. So the question is it just like the ocean then, that an enormous amount of soil carbon is being turned over simply through lysis due to phage? Or is it more because of predation from protists? Or is there a lot of osmotic shock? All of these different ways are ways that microbes can die. And maybe you don't dwell on this so much, but I think it's fascinating. Lots of different ways that they can die. Doesn't matter. Maybe it does, maybe it doesn't. So we focus mostly on phage. And we're seeing an enormous amount of mortality, particularly in the dry season in our grassland soils. And I should mention, these are just only a couple hours north of here in Hopland. And we see that particularly in the moments of the year when there's a huge pulse of CO2 are moments when we think we have a fair amount of phage activity. And we see up to something like 45% of bacterial death could be due to phage. We're still struggling quite to know which of these lower lines we're on because we don't really know the burst size to assume for a phage host interaction. That's not something we can really get out of a metagenome. So we're trying some other ways to kind of estimate that, but that would help us back out the number of virions to the number of host cells. Okay, Lest you think that I'm really focused only on bacteria, I want to talk about fungi. And in particular, our vascular mycorrhizal fungi, who are so important. You probably know this, are associated with a large number of the plant plants. So important for nitrogen, phosphorus, water transport. They, we've recently shown, can really help rescue not just the plant, but the surrounding microbiome when there's water stress. And we, with another grad student this past year, we did a little kind of back of the envelope calculation, which showed that the total length of hyphae in soil, AM hyphae, sorry, this one is AM hyphae and EM, so ecto as well, is 2 orders of magnitude higher than the length of fine roots. So if you just think about the sort of volume that they're exploring and adding carbon to and pulling water and peak from, it's just infinitely, not infinitely, it's substantially more volume than is explored by roots. So they're really, really critical that we think about them as an extension of the root zone and an extension of the rhizosphere. So we've done a number of studies on what we call, and I think lots of people now call it the hyphosphere. So the zone surrounding these little fine threads that are extensions of the fungal body. And so in that, sorry, it's so small, but that top left image there, that's an SEM and a nanosims isotopic map, which shows you that the hyphae that had been connected to an isotopically labeled plant is quite enriched itself. It's in direct connection right there with a bunch of kaolinite particles. That is the birthplace, in my opinion, of mineral-associated organic matter. That organic exudates coming out of this fungal hyphae, and they're being plastered right onto those mineral surfaces. We also see when we do a separation of the heavy fractions, so the mineral component in soil from the more particulate and organic components, a lot of carbon that has been derived from a plant has flowed through roots, through hyphae, is ending up on those heavy surface, the mineral surfaces. So they're really acting as an extension of the root system, and the pumping of carbon below ground is not just through a root system, but through this greater network of hyphae. We then just recently did a neat study, and this is in collaboration with some great partners at the JGI, to actually generate a metagenome of that tiny zone, that hyphosphere zone surrounding those little, 10 micron wide hyphae. And this is I think it's going to come out next week, which is kind of exciting. Work led by Aaron Nuccio, which showed there's really just a whole food web of interactions going on that we can infer based on who's getting isotopically enriched surrounding these microbial hyphae. So it's a number of organisms who have the proteases and other kinds of degradative enzymes that can help them break down exudates. And we think these are actually hyphal exudates. We have organisms who have the ability to be predators, and we're not sure if they're actually predating on the fungal tissue itself, or if they're predating on maybe a bloom of microorganisms, other bacteria, let's say, that are growing around them. But there's a lot of the different Mixococca and other kinds of organisms that are just known predators that are extremely isotopically enriched surrounding these hyphae. We saw fibrobacteria that we know have genes for the AMF necronas decomposition. They have a bunch of the chitinases that you would want if you wanted to break down fungal tissue. And then amazingly, we saw a lot of enrichment in ammonia oxidizers. That is not who I would have guessed would be enriched in this zone. And I will tell you, they are not abundant, but they are isotopically enriched, which suggests that somehow there's maybe urea or some sort of loop that's happening so that these organisms are getting 13 CO2 because they're autotrophs that is being decomposed by some other organism, and they're taking that up and taking advantage of being in the midst of this food web, as you will. I don't know if that's a little bit of a stretch of what food web is really meant to mean, but all of that is made possible by, again, tracing the isotopes, but also using metagenome assembled MAGs or metagenomic analysis. So that, again, will be coming out soon, and it's kind of an exciting foray into, for me, a habitat that we had just only started to think, only started to give a name to. All right, so let me give you one last story here, and this is about soil minerals, and then I'll kind of make it a little bit of a big jump to talking more about our applied work, kind of more global scale analyses. So again, I think I said this at the beginning, if we could get CO2 out of the atmosphere and park it in the ground, that would be ideal for lots of reasons. Where in the United States would we want to park that carbon? Is there anything fundamental about the mineralogy of our soils that suggests certain places are better able to hold carbon? And I mentioned earlier the notion that you should focus on soils that have low carbon to begin with. There are limits to, I think that that's true to some extent, but there also need to, there's some caution. But minerals that are what we call poorly crystalline or highly weathered, short range order minerals, tend to be much, much better at holding onto carbon than much younger minerals that are just freshly have been formed. So if you look at this map here, it tells you where you might find some of these poorly crystalline minerals, and some in the red, those that you would find simply because of rock weathering. These are somewhat older soils, but also to other drivers that have to do with cation availability and aluminum iron interactions. So you can see there's some real hotspots. It's not evenly distributed across the US. So these are, first of all, places where we might think about, if we're being purposeful and trying to put carbon in the ground, we might think about trying to look primarily at these situations where there is more ability of the mineral component to hold on to that carbon. At the other end of the scale, we've done a number of more manipulative studies in the lab to simply look at when we expose different mineral types to a growing plant, what tends to hold those rhizosphere exudates, that necromass, most quickly. And what we see is that there are big differences between kaolinite, iron oxides, quartz, and native minerals being a mixture. And a lot of that is actually driven, or at least correlated, with the fact that there are different microbes live on different mineral surfaces, right? That's kind of makes sense after you say it, but I would not have expected it to be blunt. But we see very different populations on quartz than we do on iron oxides. And that one very likely has to do with the amount of electron exchange that's going on with those surfaces. Those habitats are providing different resources for those organisms. But we see different resulting biochemical composition as well. So not just different organisms, but the results of their decomposition. So we see that in the rhizosphere, there's a lot of actually fast turnover on those minerals. So a lot of sorption happens to minerals quickly, but a lot then just comes right off. And then again, in what we call the detritosphere, or the area that's further away from growing roots, we see carbon tending to accumulate in a more slow, sort of sedate fashion. We've also, that was done in the lab, so out in the field, we've also looked at the same sort of thing. In this case, though, we didn't control the mineralogy so much. We simply added that 13 CO2 label above ground, and then we tracked it over the course of 2 years. And we looked at how much of that carbon was ending up, again, on mineral surfaces and some of those tight associations. If you look at the very dark brown in the pie charts, that's what we call the heavy fraction. And over time, it's declining, which makes sense. So that slug of carbon that got put below ground is slowly being decomposed, and some of it's being released back to the atmosphere. But I think what I found most striking about this study is that we found even 2 years later, 23% of the original slug of carbon was still there, and all of it was associated with a mineral surface, suggesting that's really the place that carbon gets stuck, that really hangs on tight. So I thought I would just mention, we're trying to get a better handle on how it hangs on. I think at the beginning of the talk, you heard me say, it's not just because it's recalcitrant, it's not just because it's aromatic or that you've got a lot of lipids or something like that. We're really trying to come up with a new thesis on, okay, then what is it other than it's mineral associated that makes organic matter stick in soil? So what we are in the midst of doing is developing a new technique to use radiocarbon analysis on single compound, or at least compound classes. And this is just some of the very preliminary data where we separate out lipids from proteins from acid residual, so sort of whatever's left over. So taking that whole mix of organic carbon, which is just incredibly complex, and try to divide it into different pools, And I think that you can see just that these data points are separating a little bit, and they're separating even more so with depth, suggesting that there are different residence times, different turnover times of the protein pool versus this acid residual versus the lipid extracts, and getting a better understanding on how those different functional classes operate is really important for how we model carbon persistence. Okay, so I'll just say, as I wrap up the sort of fundamental science part of the talk, we see a lot of evidence that deep-rooted plants and particularly fungi, and I want to give a shout out in particular to the AM fungi, but also these EPS compounds. All of those are really what I think of as the important knobs to be turning if we want to be putting more carbon in the ground. We also need to be careful, though, about how we're making our measurements and how we're analyzing the data, because there can be some statistical artifacts which make it look like we're increasing carbon when really it's a little bit more gray. And so as we are thinking about large-scale efforts to establish financial incentives for what we call carbon farming, for planting switchgrass or miscanthus or other deep-rooted grasses, we really need to be not just thinking about the dollar signs and the crediting that could be going on, but really still focusing on the mechanisms of what makes carbon stick. Okay, so I want to make a quick switch. Yes, did you have a question about stakes? Let's stay with that for one more minute. Yeah.
So on the transporting carbon to mineral surfaces. So what is the limiting factor? Are there plenty of mineral surfaces, and really you just need to select for these sort of enhanced mycorrhizal symbioses, or would it be helpful to have more mineral surfaces available?
Yeah, I don't know that we really could increase or decrease mineral surfaces other than the types of mineral surfaces. So focusing on situations where you have more of those poorly crystalline mineral surfaces might kind of accomplish what you're suggesting there. But what I will say to answer the first part of your question is we're not limited by mineral surface area, at least not in the microscopy perspective that I have from looking at organic matter mineral interactions, whether it's in the SEM or the nanosims. There is tons of open space. There's no organic matter on many, I don't have a number off the top of my head what percentage, but a large percent of mineral surface area is uncolonized by either a living or a dead or organic matter, residue of organisms. Now, obviously, when minerals get packed together into aggregates, that puts some amount of surface area off limits. But still, my sense is that we're not actually reaching that saturation asymptote in many soils, that it's available. The sponge is not full yet. It's about getting the carbon in there in the right way. Yeah.
Thanks.
All right, I'm going to dive through these last ones kind of quickly because I want to leave a few minutes at the end for questions. So as Corinne mentioned, I feel like I'm doing a second PhD in learning so much about carbon dioxide removal, sort of the activities that we need to take advantage of to not only help us get to a net zero economy, but to truly go negative, to pull CO2 out of the atmosphere so that we end up at what we like to call net zero, ideally by 2050. And so Livermore, in particular, something we call the Livermore Carbon Initiative, has been doing a number of assessments of carbon dioxide removal. We did one that was called Getting to Neutral. That was published just before the pandemic. And that was just for the state of California, and looked at all the different ways the states could pull CO2 out of the atmosphere and what it would cost, essentially generating a cost curve. And it's amazing the impact that that report has had with a lot of the legislation, which is NAT, and some of the governor's initiatives that were just announced this year were initially sort of sparked by that report. And I think the state is actually on track to achieve its goal. It's going to require obviously an enormous lifestyle shift from many people, and shifting towards agricultural practices, forestry practices, municipal waste transport and storage. All of that is part of a puzzle that can conceivably actually get the state to net zero. We've done subsequent analyses for Microsoft, and we're now doing one that we call roads to removal for the whole of the United States. And that's what Corinne and I are collaborating on with a lot of other scientists We're looking at both soils and forests as the sort of natural solutions, but also direct air capture, geologic storage, and biomass conversion. We're trying to then really link all of this together, because a piece of land that's growing a biomass crop, you could conceivably be generating biomass for some electrochemistry or gasification process above ground while trying to also store carbon below ground. That might be a little too much to ask to really be gaining carbon in both, above and below ground, but conceivably it should be possible. We also have a real overlay of environmental justice on all our analyses, because I think in a lot of the previous accounting that had been ignored, that we can't just willy-nilly use this land for something that is not supported by the local populations who live there, but there are also potential job benefits for a lot of these strategies. So we're trying to create maps that at the county level have an assessment of both the availability of a particular kind of CDR and the cost. So this happens to be a biomass map, looking at where we have high biomass density and where we have low transport cost. We're going to do the same thing for DAC and for geologic storage, and I could go on and on. And again, it's a really huge team. Folks all over the country are helping, both from the economics to the modeling to geologists who are telling us about new storage reservoirs. I think I'll go through this pretty quickly. We're looking at weighing different strategies in soil that range from cover cropping to the deep-rooted perennials to simply set-asides, putting land into CRP, conservation reserves. We're looking at shifting cropping from current annual crops to more perennials. This happens to be a map of what would happen if you increase the amount of cover cropping, and trying to also, I should say, not just look at the carbon below ground, but look at changing the amount of greenhouse gas emissions. So if you have less N2O being produced or less methane, we're counting that. So in this particular analysis, we've looked at if you're switching land from corn, soy into switchgrass, what does that affect? How does that affect N2O emissions? And I want to be clear that this is done in collaboration with actually one of the other BRC teams, led by Bruno Basio and Phil Robertson, who are part of GRLBC. So obviously a lot of this land needs to stay in corn soy if we want to feed people. That said, we've estimated there's something like 10 million acres of land that could conceivably come out of corn production if we have less need for ethanol that goes into transport vehicles. And that, again, as we electrify our transport sector, could conceivably be possible. Some of that may still be needed for sustainable airline fuel, but I'm hoping you guys are going to solve that problem. And we will actually have somewhat less pressure on land in this country. And we can imagine some of it going into a more deep-rooted perennial biomass crop, or even a conservation situation. So this is a really complex analysis, but essentially what we're looking at is, a lot of people grumble, well, soil carbon isn't permanent storage, right? It's not durable like putting something in a geologic storage well. And so we looked at, what if you do just temporary storage in soil? Does it make sense to do a soil management project here and then go do one over there and then go do one over in the other part of your county? Or does it make sense to actually maintain the soil carbon that you accrued in site number one and then start in another site and another? And essentially, long story short, we're showing it does really make sense over the sort of 75-year time span before we get to 2100 to maintain the soil carbon that we've accrued in the site number one. So let's say you plant switchgrass or miscanthus, or you put land into a conservation reserve program, and there's an initial kind of bolus of carbon that you accrue. Even if that land isn't continuing to accrue more and more, it makes sense financially and from the kind of global warming potential aspect to just keep that carbon where it is and not just keep moving to new sites and re-implementing that strategy. We've done also an analysis of where biomass is available and where it could be processed. So all of those lines here are, just one second, different types of biomass, and then where a likely processing facility could be. And down in the lower left is our initial analysis of geologic storage. Obviously, there is a lot more optimism about where we could do geologic storage than there's actual, boots on the ground in effect. But we are optimistic that that is a real solution. That if you are gasifying, that you're always also doing carbon capture. Even if you're doing DAC, you're doing carbon capture that is then put into a geologic storage situation. Yes.
So in the previous slide, when you were giving the two alternatives
Yeah.
about either doing the sequestration in one place and maintaining it
Yeah.
versus setting up these sort of smaller time scale, shallow things all over. What's the fallout from that? Are you locking a particular site for sequestration? Who would give up their land for that? What would be the benefit to them?
So I think a lot of this merges into economics and into spaces that I am less well trained in, we'll say. But working with Jérôme Dumortier, he's looking at both the tax benefits, but also land management, financial benefits that folks get for putting land into CRP, or it's sometimes called EQIP. And often that is marginal land, which is literally not financially viable to be doing sort of intensive agriculture on. And the idea being there that's the trade-off for putting land aside and out of constant production. That's the financial reward for the landowner. Some of that is consistent, congruent, is that the right word, with actually producing a biomass crop, as long as you're not plowing and you're not doing kind of intensive manipulation of the soil. Yeah. So let me just end with my last data slide, where I just want to give you some evidence of how we're trying to look at environmental justice. That's a huge space. And again, it's why I'm saying, I feel like I'm doing a second PhD, and this is like a really new area for me. But we're trying to look at what existing data exists about both pollution in groundwater. So that in this one, first on the left-hand side there, you're looking at nitrate in groundwater, but also air pollution, thinking about where funding and new jobs are being potentially being made available as we switch to these different economic incentives for doing direct air capture or doing geologic storage. But also looking at who owns farms, who is the labor on farms, and on the landowners and their relationships with their labor force. So we're trying to think about what if we were to give incentives for specific counties or specific parts of the country, who would benefit and who would lose, not only in terms of jobs and land ownership, but the health of their air and water resources. So all of the mapping that we're doing is not just of the sort of physical and both above and ground realities. We're trying to have this sort of overlay of the human that impacts as well. So that's what we're calling our environmental justice analysis. I want to end here and make sure you have time to ask a question or two by thanking not only the work that funded the sort of whole first half or 3/4 of the talk, which is the Microbes Persist SFA. It's a soil microbiome SFA funded by PER, but also a project that we had from PER looking at sustainable switchgrass production, and then the Carbon Initiative, which is kind of the center that's supported funding from FECM and RPE and BETO for the CDR work that I showed at the end. Thanks. Yes.
I was very intrigued when you said earlier that 8% of the microbiome is active. What's the other 92% doing?
I would love to know. Okay, so let me say this. That is true. It was actually about a month ago when we had that first rain event. Do you remember we had this sort of weird kind of Pacific hurricane, and in September we got this sort of freak storm? Right after the first rains happen in annual grassland, there's this huge pulse of CO2. And we call that the Birch effect. You can lose a large amount of the carbon stored over the course of the year. At that moment, and we've measured this year over year, only about 5 to 8% of the total microbiome is active. That said, they've just experienced months and months of incredibly low water potential and no ability to move and osmotic shock and all sorts of things. So that's probably I'm being a little, choosing the worst, I don't know if it's the worst, but the lowest percent activity at time of year. If you were to go and measure maybe in January or even into March, when those, grasslands are starting to, really green up, it would be up in the 60s. That's still though, what is the 40% who's not active then doing? We're actually doing a study now that goes throughout the whole year to try and really map, which populations are abundant and active at what time. It could be the organisms who really love the wet up, the week after wet up in the fall, are just sleeping, or it's just, things are not ideal. That's not their niche when it comes to next March. It also could be, there's always, methanogens. There's a bunch of anox, firmcutes and other organisms who are truly adapted to low oxygen conditions. That's not their sweet spot if you're thinking about, when we've got a fully oxygenated rhizosphere and lots of carbon being produced. So I think that it's a little mystifying, but there does seem to be a lot of different niche, what we call niche differentiation, that not everybody's happy all at the same time.
So what you're hypothesizing is that they're not necessarily dead, but they're in some sort of persistent state.
I do
or
think that.
latent state.
Yeah, I think that it's a little bit of both. I will say that there's been a couple of studies, and I wish we could get more, but the methods are hard, that show there's quite a bit of what we call relic DNA or eDNA out there in soil. It's probably everywhere. It's nucleic acids, both DNA and RNA, which simply persist, which again, I was taught, no, that stuff will get chewed up by a nucleus, right away. But turns out, no, there are lots of different absorption mechanisms. A lot of organisms use nucleic acids to make EPS and to create kind of habitat for themselves. So there's some proportion, maybe 15–20%, that's literally non-cellular nucleic acids. But then I think there's another crowd that's dormant, sleeping, sporulate, just in some other state, not interested in taking up 18 water and not dividing, because those are our metrics of alive. And I recognize you can be active without actually growing and dividing, but that's my definition of it. Yeah.
Okay, two things. First one's really quick. Thank you for studying fungi.
Oh.
I know
Smiles.
they're really hard to study, but they're so they're so important logically.
Yeah.
And then secondly, I've worked in a couple different native grasslands, and so I know the diversity per square meter can vary from anywhere from 5 to 10 species down to 30 I've seen in Uruguay. And so I'm wondering what you know of the land use history of the places from the first studies that you talked about when you were comparing switchgrass to annuals, what you know about that, and if that could potentially explain some of the differences that you see. That's
Yeah, so what I know about the land use history of that area is that it lost somewhere between half and a meter of topsoil during the Dust Bowl. I can't help but think that that was a complete reset of soil formation, of the biological diversity, of just everything about the soils. I don't actually know how much was lost from that particular site. We have land records in that site going back to the 50s, because it's actually owned by the University of Oklahoma. But what you're pointing out is incredibly important. And I'll just say in all of those other sites around the US, we don't always have that, those land records. And we certainly don't have records of microbial communities. But I think there are many, many things that could have happened 80 years ago that have an effect today. And if I didn't happen to know the Dust Bowl that occurred, I just wouldn't know that the soil that I'm seeing as an A horizon originally was a B, or I don't know if you're a soil scientist, but these are the different layers of soil. It was originally not where roots are growing. So really it's a very young soil in that sense. So there's always more to be learned from just site records and land, talking to people who actually live on the land. Yeah.
Kind of going back to what you showed about the AM food web situation.
Yeah.
I know glomalin has been something that's been applied to be really important for soil structure and is posited to be produced by AM fungi. But thinking about the microbiome situation, it kind of raises the question: or I guess my question to you is, do you think glomalin might be related more to the EPS produced by the microbes that are on the AM, or is the glomalin something that might be attracting those microbes to come do their thing?
Right. So the first thing I will tell you is we think glomalin doesn't exist. Sorry. There are a lot of papers on that, and they were mostly coming out of one particular lab. And then there was some very key work that showed that molecular moiety is produced through the boiling and very intensive extraction procedures used to define it. We think it is a measure of what comes, it's produced as opposed to pre-existing. That said, I think the concept is very real, and we have lots of evidence to suggest carbon flowing through fungal hyphae is being exuded and being taken up by all these other organisms. And we actually see some evidence of it getting onto mineral surfaces. We think that that may be different kinds of peptides. We don't have an answer really of what it is. I think there is something that fills that conceptual niche of glomalin. It's just not called glomalin. Does that make sense? It's going to be like an EPS, most likely. And I just don't know whether it's going to be polysaccharide or a polypeptide.
Thank you very much.
Yeah.
Do you think there's any space for these direct carbon capture mechanisms, like converting CO2 to formate, in conjunction with using marginal croplands? Is some way of synergizing these two ideas?
I think there probably is. I'm looking at Corinne because she's in charge of the cross-cutting aspect of our analysis. I don't know that we've really thought about that one per se. We have thought a lot about where biomass can come from. And in certain parts of the country, it needs to be purpose grown. So it's brassicas, canthus, cane, whatever your flavor of the week is. In municipal areas, it's often more from municipal waste. In the state of California, what we calculate is the money that we spend in moving just our organic garbage around. If that money were put into carbon storage below ground, it'd be the same amount of money, and we would be storing carbon. So I think that there are ways, yes, to do things on the same piece of land. It just depends if it's an agricultural field or a forest or Berkeley or Emeryville. I mean, there's kind of a different answer very much depending on what hectare you're talking about.
Totally grow feedstocks with the goal of sequestering soil carbon, and then also take that material and convert it to whatever you want. And if you're doing a process that produces CO2, let's say maybe you're still fermenting to ethanol and you get a nice pure CO2 stream, by all means, you could convert that too, or you could geologically sequester it. And I don't know if we've gotten super deep on the geologic sequestration versus conversion of the CO2 question. A lot of times it is kind of like,
yes,
put it underground.
And the geologic storage, the financial piece there is still really tough to figure out. It has to do with transport and whether pipelines already exist and whether permits already exist and whether you've got an EJ group that's going to be really upset if you do it because they think you're really just trying to do EOR. And there's just a lot of stuff there that is yet to be figured out on what the true cost is going to be to do geologic storage. I think we have actually slightly better sense of the benefit of actually a precursor molecule that goes into the bioeconomy.
It's easier to model.
Yeah, not that it's easy, but it's easier.
Can you please tell us a bit more about the labeling patterns, the talloys, from the microbial population that you sampled that came from the plants fed on label 13 CO2? What are the lessons from those observations for engineering the microbes for improved carbon sequestration?
So we've got labeling of a plant. It's an expensive CO2 space. And we're tracking that 13C so that it's going to be incorporated into the tissue of the plant. 15 to 30% of the photosynthate the plant fixes is exuded as rhizodeposits. That's a catch-all term that means exudates, sharing with fungi, sloughed off cells, everything that's happening while the root is still alive. So there's a big amount of the plant's effort that's literally just being shunted into the below ground community. And some of that, maybe 10% of that just gets directly sorbed onto mineral surfaces. So that's the pathway. what is it that we could enrich in the microbial community that lives in the rhizosphere? Well, first of all, I think that if the AM interactions and ECM, but in grasslands, it's going to be mostly arbuscular mycorrhizae. If those are even more robust, we get more of that fungal network. We get more of those organisms exploring soil and getting in contact with mineral surfaces. So one of the things that I've talked to with the folks who are doing the CRISPR engineering of plants is there's something about the arbuscule that, so what's transferred between the plant and the arbuscule is actually a lipid. If we could engineer something to make that process more efficient, would that actually create more biomass and better flow of carbon out into the soil? At some point, we don't want the plant being damaged, right? If it's just a shunt for carbon into the soil, it still needs to, keep itself going. But that is one place where I thought about, kind of co-engineering of a plant microbe symbiosis. I will say, I personally think if we're going to do some kind of engineering, in my mind, it makes more sense to work on the plant. We control plants, we produce seeds, we disseminate seeds across the world, across landscapes. We're not good at distributing plant beneficial or soil beneficial microorganisms across landscapes whole scale. I remember seeing a talk at the DOE once where somebody was talking about all the methane being produced as permafrost as it warms. And George Church stood up at the end and he said, well, why don't we just engineer them to stop doing that? And we'll just, take care of them. And I was just like, I just remember feeling dumbfounded, like, okay, I believe you maybe could do that in a culture in the lab, but how do you then, put that out on a landscape scale? And how do you make sure that there aren't like incredible biosecurity concerns about, introducing that organism into the world? So I think that we have more control, even though we have problems still with engineering plants and distributing those across landscapes that then partner with indigenous microbes, that organisms that are already there, or maybe come with you probably know about Pett-Ridge and their nitrogen fixing friends. So scenarios where there's sort of a close partnership and you're delivering a seed that's been kind of previously inoculated, as opposed to actually taking wild populations that you try to enrich in the soil. If somebody comes up with a way to do it, that's amazing, but I don't see that as sort of top of my list. Well, thank you again, everyone.
One more time.